I want to thank Radham and the organizers for inviting me here. You guys are a fun group of folks, and I'm having a good time. All right, so I want to talk to you today about translation, which would seem to be a pretty arbitrary topic. But believe me when I tell you that at the forefront, at the edges of modern biomedical pain research, this is the biggest story in town. This is the biggest hill that we currently need to climb. And translation is really, really tough. And of course, when I talk about translation, I'm talking from going from one animal to another. The difficulty of this was noted a long time ago by Arturo Rosenbluth, and who basically pointed out that the best model of you is you, right? And even another human isn't really a very good model of you. But of course, as a practical matter, we can't do experiments on you. We, at the very least, can only do experiments on other people. And we can't even do that. And we could have a whole hour-long talk where I could try to convince you the reason why experiments in humans are not real experiments. Now, I'm not saying that no data can be obtained from humans and you can't learn things from humans. You can, of course. But to do true experiments that provide causal information, right, where you can say this causes that, you cannot do this in humans. You can only do correlational experiments. To do causal experiments, you need to go to animals. And of course, as you'll see, we've opted to go for rodents because they're small and cheap. And from rodents, we want to learn things about humans. And the question is, can you do that? How well can you do that? Of course, you can do it, right? There are many people like me that do this all the time, and I've been doing it myself for 20, 25 years. And in that time, I have come up with what I think is a great irony, or at least a pair of ironies that I want to talk about in this talk. And the irony has to do with the biopsychosocial model. So I assume everyone in this room is familiar with this. The assumption that we all make about the biopsychosocial model and translation is that at the low level, the bio level, things probably should translate pretty well. Whereas when you go up the biopsychosocial model into the psycho and the social levels, you would imagine that translation from animals to humans would become much, much more difficult. I'm going to try to convince you that this is wrong and actually backwards, which is very strange. And we'll see at the end if you believe me or not. So suffice it to say, we've come a long way from the first guy who took a hard, long look at how pain mechanisms work. And this, of course, is Descartes' boy. He had it wrong, not completely wrong, but obviously Descartes didn't have it right. We've learned some things from them. So we've learned what's going on in the spinal cord, and we've learned how pain information ascends up to higher levels of the brain. Especially these days, we're now able to go and really take a long, hard look in great detail at the microcircuitry inside places like the spinal cord. And of course, with imaging technology, we're now also able to figure out, if not how, at least where pain is processed in the brain, even of humans. And yet, with all this knowledge, and it is impressive, right? A lot has been learned, but despite that, we are still faced with a major translational problem whereby almost nothing ever gets approved. We do, huge drug discovery work. Things go into, preclinical trials in universities and in drug companies. And then we're faced with the so-called valley of death, where of all the compounds that look like they should work, only a few of them might work, and at the end of the day, only 1 in 10,000 gets approved. If you look at the reasons for this translational failure, you learn something very interesting and very surprising. So if you look at that pie chart, you see that the #1 cause of translational failure, that is to say, a drug looked good enough in animal models that drug company executives were willing to give the green light to spending $10 to $100 million on clinical trials, and then the drug was found out not to be efficacious in humans. That's what lack of efficacy means. It worked in animals. It really looked like it worked, but the clinical trial didn't show that it worked in humans and the drug died. Okay? That is obviously a major, major problem. And people that I go to conferences with have been gnashing their teeth and wringing their arms about why this might be for quite some time now. It strikes me that there's a limited number of explanations for this translational failure that have been proposed. And here they are. On the one side, the preclinical side, a lot of people have talked about the problems with the existing animal models. With their generalizability, possible species differences, and the dependent measures we use in our experiments. And then very recently, there's this thing that's going around the world like a virus, suggesting that science doesn't work at all. It's irreproducible. No lab can reproduce another lab's findings. On the other hand, some people have placed the blame for poor translation on clinical trials. That the idea there being that these drugs actually really do work in people. It's just that the clinical trials are increasingly unable to show it. And that's for a number of reasons that I've listed there. So when I look at this list, I've noted to myself that my laboratory has actually worked in a number of these domains and provided evidence that has contributed to how people think about this, and I've highlighted those in blue, and I want to go through them in the first part of this talk to tell you what the state of translation is, why we're failing, and how we can do better and are starting to do better. So let's start off with the issue of genetics. This is where I started my career and have spent a lot of time finding pain genes. The first thing to say about genetics is that of all the species that we could have been using for pain research, we've actually settled on 2. And this has actually been a recent phenomenon. Back in the 60s and 70s, there were other species that were commonly used in pain research. But now it's all rats and mice. You can see that it's actually a lot more rats than mice, which is a strange thing about pain research. In biomedicine overall, the mouse is the #1 species. In pain research, it's the rat, as it turns out. But these two have a huge lead over everything else. So of all the genetic diversity available to us on the planet, we've actually settled on 2 rodents. This obviously is a problem right off the bat. What makes that problem worse is that even within those 2 species, the vast majority of pain research is done on one kind of mouse and one kind of rat. Right? So if you're using the mouse, almost everyone is using C57 black 6. I actually think the real proportion of black 6 in the literature is higher than that. It's just that no one knows how to spell C57 black 6. So when you do a PubMed search, you don't catch all the possible permutations. And in rats, almost everyone uses a single kind of rat known as Sprague Dawley. So again, we've taken this genetic diversity that we could be sampling and we've reduced it down and settled on 2 proxies of all rats and all mice and all people, right? The problem, of course, is that strains are different from each other. And so this is the study at the very beginning of my career that probably got me tenure. And I know you can't see what's going on here, but it doesn't matter. The point is that you're looking at the responses of 11 mouse strains on 12 different pain tests. And the point is simply that in every case, the landscape is rocky, right? Some strains are sensitive, some strains are resistant, some strains are average. It really very much matters which strain you use. And it's actually worse than that, because in addition to there being quantitative strain differences, there are also qualitative strain differences. And I'm going to give you an example. So what you're looking at here is the ability of a compound called CGRP, calcitonin gene-related peptide, to increase pain sensitivity in a mouse. And it increases the pain sensitivity, so it makes animals withdraw from thermal heat faster. And then the CGRP metabolizes away and everything goes back to normal. Okay? So if you looked at data like this, you would conclude that CGRP had something to do with heat pain. Right? The problem is if you do that exact same experiment in this strain, C57 black 6, you don't see any effect of CGRP at all. And it doesn't matter the dose. Right? There is no dose of CGRP that produces an effect in C57 black 6. And if you do the same experiment on 12 different strains, you find that half of them CGRP works and half of them CGRP doesn't. So what's the conclusion? Is CGRP involved in heat pain or is it not involved in heat pain? I don't know. And the problem is that the scientific literature is not as big as you think, right? There's probably only one lab that would ever do this kind of experiment. And if they happen to be using a strain on the left, they would conclude one thing, and everyone would believe that that was true. And if they happen to be using a strain on the right, they would conclude something else, and everyone would believe that's true, and no one would ever know. Right? And this is a major, major problem to this day because obviously for reasons of cost, we only do experiments on one strain at a time. And then everyone just believes that the result generalizes to all mice, but it doesn't. The same problem is there with sex, although this is something that gets people even more interested. Ultimately, the problem is that we are using the animal on the right to stand in for the animal on the left, right? And the problem isn't so much that it's a rat. The real problem is that it's a young male rat of a single strain standing in for a middle-aged woman with, complex genetic background. How bad is this situation, using male mice to stand in for women? And of course, women, as I'm about to tell you, are the real clinical problem, right? And here's data to show that. So what you're looking at here is data from a review of epidemiological studies that I published in 2012. And all these epidemiological studies were different. They were measuring different pain types, and they had completely different definitions of what chronic pain was. But it doesn't matter, because in every one of those studies, the definition for men was the same as the definition for women. So all I've done is I've subtracted the male number from the female number and plotted it there. And you can see that in every case except one, women were more likely to endorse that they had chronic pain than men, again, however the definition was derived. And on average, it was by 5 to 10%. So that's a really big excess of clinical pain in women. And presumably because of that, if you go and look at patient roles in any kind of pain practice, right? Any kind of pain practice, you come up with numbers like that, where 70% of the patients, of the clients, are women. So that's the clinical problem, right? 70 to 30. So what are we basic scientists doing? Here's what we're doing. So this is a survey of papers published in the flagship journal in the field called Pain over a 10-year period. And I've shown you the number of studies that only use male mice, that only use female mice, that use both sexes but don't talk about it, use both sexes and didn't find anything, use both sexes and found something, and an amazing 7% of studies that didn't even say what sex of animal they used. And you can see that the vast majority, 79% of these studies, just use male mice or male rats. Right? Interestingly, I wrote an article that came out in the journal Nature last year, and they asked me to update this number. So this was from 1995 to 2005, and they wanted me to do it from 2005 to 2015, and I told them, no, that would take too much time, but I'll do it for the year 2015. And so I did. I went back to the journal Pain. I opened up all the papers using mice and rats in 2015, and I counted up the same proportions. And guess what the proportion of studies was in 2015 using male mice only? It was precisely 79%. No progress made over a decade. So here's the mismatch, right? Why does it matter? Because you might say to yourself, well, who cares, right? I mean, unless the biology is different, we can use males to stand in for females. That should be fine. So I'm going to try to convince you over the next few minutes that you are making such an assumption at your peril. Let me start off with the cautionary story of a drug called Morphodex. So what you're looking at here is the ability of a drug called dextromethorphan to potentiate analgesia from morphine. And these are our data, but you can go into the literature and find like 100 studies that show that. This is a big deal in the late 80s, early 90s, and people got very, very excited about this finding because dextromethorphan is known to be a perfectly safe drug. And the reason we know it's perfectly safe is that it's the active ingredient in cough medicine. And cough medicine has been sold over the counter for decades. If there was any safety problem with cough medicine, we would know it by now, right? So the idea is you can take a perfectly safe drug, add it to an admittedly very unsafe drug, morphine, get more analgesia, well, therefore you could use less morphine, right? And everyone said, boy, what a lovely idea. But here's what they didn't know, because every single one of those 100 studies in the literature used male mice or male rats only. If you do this study in female mice, there's no interaction at all. At no dose of morphine, at no dose of dextromethorphan, do you get any potentiation of analgesia. And maybe, just maybe, that's why the resulting $30 million clinical trial completely failed. So what you're looking at here is Morphodex, a one-to-one combination of morphine and dextromethorphan, and you can see that the Morphodex doesn't work any better than the morphine alone. This drug failed. The drug was shelved. Of course, clinical trials are by law done in both sexes, right? Men and women. And so I emailed these guys and I said, so I now have reason to believe that there's a major sex difference in the ability of dextromethorphan to potentiate morphine analgesia. Did you guys note any sex differences in your clinical trial? And they said, I don't know, we didn't look. So they had the data from both sexes, but they never actually bothered to analyze it to see if there was a sex difference. And so I said to him, I said, well, for God's sake, man, go look. and he said to me, and I quote, I don't know, I'll have to ask the lawyers. And indeed came back to me a week later and confirmed that the data had been sealed and the drug company refused to open the data. They simply didn't want to know. The quote from the lawyers was, we've moved on, leave it alone, right? They simply didn't want to know the answer. So maybe Morphodex worked in men and not women, but we'll never know. It turns out that this same thing is probably happening again right now. And don't worry, I don't want this to scare you. I'm not going to walk you through this diagram. I only put it up there to show that one of the major advances, one of the major new concepts in pain research over the last 15, 20 years is that we used to think that, you're looking at the spinal cord, by the way, we used to think that in the spinal cord the only cells that mattered were the green ones, the neurons, right? And everyone was studying the biology of those neurons. But now we know that the astrocytes and especially the microglia are playing very important roles as well and are just as involved in the signaling that leads to the opening of the gate and the ascending pain information. The microglia are just as important as the neurons. The problem is, as you might have imagined, all this work has been done in male mice, and we now have reason to believe that the entire microglial involvement in the spinal cord is entirely male-specific. Now, I'll show you just a little bit of that data, and again, I'm not going to spend too much time walking you through the data, but suffice it to say, you start off with animals that have a certain sensitivity to mechanical pain, you give them an injury, they become allodynic, right? It's important to note, as you can see if you take a look at that graph, that the amount of allodynia does not show a sex difference. Males become allodynic and females become allodynic. The sex difference is shown when you try to reverse the allodynia with a drug, and you can see that's working very nicely in male mice here in a dose-dependent manner, but it's not working at all in female mice. And what you're looking at here is the glial inhibitor and antibiotic called minocycline, but you can get the same effect with fluorocitrate, and you can get the same effect with propentophyllin, and you can get the same effect by killing the microglia, and you can get the same effect by blocking the microglia at the beginning, in the middle, or the end. Suffice it to say, we have impaired microglia in every way that science knows how. In every case, the microglial impairment blocks the allodynia in male mice, and in every case it does absolutely nothing in female mice. And so we are working on a hypothesis that's complicated and I'm not going to spend time on now, but it's looking more and more like male mice and rats, we now know, are using microglia to process pain in the spinal cord, whereas female mice and rats are not using microglia. Instead, they're using T cells, and the T cells are producing approximately the same effect as the microglia. No sex difference in the amount of pain, huge sex difference in how that pain is processed. And of course, my prediction as of today is that any drug that is developed that works by blocking something on microglia is not going to work in women. And any drug that's developed that works on blocking something that T cells release is not going to work on men. And of course, there's no example of that yet in biology and biomedicine, but it's coming, and my prediction is it's coming in pain first. I want to move on to the real critical translational issue of how we measure pain in animals in the first place. And this, as you can imagine, is a challenge because they don't speak English very well. And so you have to infer it some other way. The problem that I and others have noticed is that it seems that pain researchers are spending their time on entirely the wrong symptom. So what you're looking at here is a big study of patients with, in this case they had back pain, but there were a number of different types of pain in this study. The researchers measured symptoms and signs from this clinical population, and the size of those dots is reflective of the prevalence of that particular symptom in that population. And so you can see that some symptoms are common and some symptoms are much less common. Here's what we measure in animals. So again, this is from 10 years worth of papers published in the journal Pain. And you can see that the most common symptom that's measured in animals is mechanical allodynia, von Frey fibers onto the hind paw. And trailing just behind is thermal hyperalgesia, which is increased sensitivity to heat beam. And only in 10% of cases do the authors even claim to be measuring spontaneous pain. And sometimes when you look at what they actually measured, you really wonder whether it is spontaneous pain. So I've color-coded those on the right, and now I'm going to show you how they map on to the situation on the left. So you can see that the things we measure in animals are barely even there in human clinical populations. And the big problem, ongoing deep pain, is something we don't know how to measure in animals. No one does it, no one knows how to do it. Mechanical allodynia is much easier, and thermal hyperalgesia is even easier than that. And that's ultimately, I mean, I hate to say it, but what it comes down to is what scientists are ultimately interested is, how do I publish papers? Right? The fact that it has nothing to do with the real world is a secondary issue entirely. People like me started pointing this out. People were studying the heck out of a symptom that isn't really very common and not really very important. Let's study the right symptom. And in fairness, people are starting to change, because in the past few years there have been any number of new methods developed that claim to measure spontaneous pain, including operant conditioning, classical conditioning, wheel running in a cage, ultrasonic vocalizations, burrowing into a tube. And in fact, some of the biggest labs in the field are turning at least some of their attention to going back to the drawing board and coming up with these new models. Our particular contribution to this is the mouse grimace scale. And we developed this basically by adapting the scale that is currently used with babies. So babies are another population that don't speak English very well. But you need to sometimes quantify their pain as well. And one of the most useful ways of doing that is to look at facial grimacing. And it turns out that mice and rats and other creatures do about the same thing. So orbital tightening, nose bulge, and cheek bulge are right off the baby scale. And if babies could move their ears and had whiskers, I'm sure they would do that too. And it turns out to be pretty easy to train yourself to use this. So easy, in fact, that we developed it for the mouse and then the rat. And now other people around the world are developing them for all kinds of species. The ones shown here are the ones where there are currently published grimace scales. And I should point out that a lot of this work has been done by Matthew Leach at Newcastle, who got a big grant from the UK government to do just that. So I'm actually kind of proud of this because as you can imagine, as a basic scientist, it is very, very rare to have any of your findings reduced to practice in any reasonable time span, right? Usually my hope would be that, well, 40 years from now, there might be a drug that, you know, I had a little bit to do with. But grimace scales are being used by veterinarians today, and it only took 2 or 3 years. And so that's neat. I would have rather cured human pain, frankly, but you know, I'll take animals. It's not bad. All right, moving on to the notion that maybe preclinical science just doesn't work at all. And this is a notion, in fact, that's been considered seriously by important people and continues to dominate news reports on science to this day. Usually, the hypothesized causes of this problem have to do with statistical issues and procedural issues. So the idea is that our sample sizes are too low, and we're not using statistics right, and we're not blinding ourselves. And frankly, there might be fraud in the system as well due to poor incentives. And I've actually published a little bit pointing out that this is a little bit oversimplified, that there's different types of preclinical research that need to be held to different standards. But why I bring this up really is because I think a lot of this is just dead wrong, that science is perfectly reproducible, or at least as reproducible as it can be. It's just that there are a lot of factors that affect results of experiments that we've simply never considered before, and I want to give you an example of that. So this is a surprising cause of what would look like irreproducibility, but it's not. What it really is that certain laboratories have a certain situation and other laboratories have another situation, and they're both right. So here's what you're looking at. You're looking at the facial grimacing of mice if you inject an inflammatory substance into their ankle and then put a video camera on their face for 30 minutes. Okay? And the scale is out of 2, so that's a pretty reasonable amount of pain. What happens here is that an experimenter goes in, injects Zymosan in the mouse, and then leaves the room, and the video camera captures the facial expressions for 30 minutes after that. What I'm about to show you is what happens if that experimenter, instead of leaving the room, sits down in a chair and reads a book for half an hour, or if another experimenter comes in, sits down in a chair and reads a book. Okay? So there's less facial grimacing, less pain. It looks like the presence of the experimenter is causing analgesia. Okay? But here's what makes this finding fun. Because it's only the men. The women in the lab do not produce this effect at all. Right? It's only the men. And for a while, we thought, well, maybe men are more convincing predators to mice because they're bigger, but I never believed that for a second. One of those women is a lot bigger than all of those men. She is. And it turns out that in the fullness of time, we found that no, this has to do with olfaction. And it turns out that the men in this room are throwing out of their armpits as we speak certain chemicals at higher concentrations than the women in the room. The women in the room are throwing out other chemicals at higher concentrations. The 3 chemicals shown there are the ones that we can get the analgesia in a concentration-dependent manner just by opening a vial of that chemical in the room. Okay? And 2 of them are very famous, androstenedione and androstenone, but one of them is more important, 3-methyl-2-hexanoic acid, because it can produce the effect at the lowest and most reasonable concentrations. And so what it really looks like here is that, yes, indeed, mice and rats are afraid of men. At least the smell of men stresses them. And the reason that human males can produce this effect is because, well, we smell just like male mice, right? In the way that counts, anyway. All right. And then the final thing I want to tell you about in terms of part 1 of this talk about how translation's going at the low level is the issue of clinical trials. Now, one of the dominant hypotheses out there is that the reason that clinical trials are failing in the present time, which they are, left and right in pain, there hasn't been a novel pain drug that's not an opiate that has been, that's not true. There's been one. Ziconotide has been approved. But other than ziconotide, there hasn't been a novel pain drug that's been approved in 20 years, except for opioids and ziconotide. And people think the problem is the placebo effect. And they have some reason to believe by looking at trials that placebo effects are getting suspiciously larger with time. But it was unclear whether that was true for pain. And so we did a little search of the literature where we took all the trials of drugs for neuropathic pain published in that time period. And we took information right off the graphs of that study. And indeed, as you can see in B, it is true, placebo responses in these trials are getting bigger over time. They used to be around a 10% improvement, and now they're around a 30% improvement. The problem, of course, is that the drug response group is not getting any bigger over time, so that treatment advantage over time is decreasing. And of course, treatment advantage is what gets a drug approved or not approved. So this is true. What everyone thinks is happening, but here's what makes it interesting, because it turns out that it's only true in trials conducted in the United States. Trials conducted in the rest of the year, the placebo effect is not growing, at least not growing significantly. And your guess is as good as mine about why that might be. It's either something about Americans, or it might have something to do with the fact that only the United States and New Zealand allow direct-to-consumer drug advertising, or it might have something to do with the fact that the use of contract research organizations, for-profit companies that run clinical trials, is much more penetrant in the United States than elsewhere at the present time. Frankly, I don't really know. But I think it would be really a good idea for people to start figuring this out. All right. So, on to part 2, and the experiments I want to tell you about here all involve social manipulation. So the very tippy top of the biopsychosocial model, the kind of thing that you would expect wouldn't work at all in animals. And we talked about this yesterday, but I'll very briefly remind everyone that this really, the social pillar of the biopsychosocial model is really neglected. There's just a huge paucity of research on the topic compared to biological and psychological. And it's really about time, in my opinion, that people started taking this seriously. Can you do this in mice? Well, yeah, actually you can, because laboratory mice, believe it or not, have a social life. Now, I know it's not as good as your social life. But they do things, even in a cage, that can be measured and altered, right? There are any number of social behaviors that you could quantify and that you could try to manipulate. It's absolutely doable. Now, whether this has anything to do with people is another story, but at least there's a place to start. Here's where we started. We started with this phenomenon that has a terrible name. It's called socially mediated contagious pain hypersensitivity. It involves a dead simple, very old pain test whose name has gotten me in a lot of trouble over the years. I didn't name it. It's called the writhing test. That's its nickname. It's actually really, really mild. And I know it's mild because the writhing behavior in this test can be completely abolished by aspirin. And anything that aspirin can take away completely doesn't hurt that bad. Under normal circumstances, we take a mouse or a rat, we inject it with acetic acid, we put it in a plexiglass observation cylinder, and we start counting writhes. Well, the students start counting writhes. In this paradigm, we added 2 dyadic conditions where there are 2 mice instead of 1. In one condition, the one writhing condition, there's a mouse that's injected and a mouse that isn't. And in the more interesting both writhing condition, now there are 2 mice that are injected. So both mice are in pain, but they're also looking at another mouse in pain. And the question is, does that matter? And of course it does, or I wouldn't be telling you this. Here are the data. Okay? So here's how many writhes you get if there's just 1 mouse. Here's how many writhes you get if there's 2 mice, but only one's injected, the same. And here's the effect. So if both mice are in pain and they're looking at another mouse in pain, there's more pain. Okay? And what makes this interesting is that that's only true if they know each other. Okay? So if they're cage mates pulled from the same cage, you see this effect. And if they're strangers, you do not. And after years and years, literally 2 years of control experiments that I won't bore you with, we concluded, and the reviewers agreed, that what we were looking at here was empathy. We were looking at emotional contagion. And this was kind of a big deal because it was the first time emotional contagion had been demonstrated in any species other than humans. Even primates, they hadn't gotten pretty good evidence yet. We now believe, I and other social neuroscientists now believe that mice are perfectly capable of both consolation and targeted helping as well. Empathy, of course, being sort of a multi-dimensional concept. And so this finding really sort of sparked a renaissance of this sort of work, which I'm very happy about. I want to get back to the issue of why you can't show, by the way, this is, I think, this is a real thing. I think it was like an art installation in the New York subway. So why is it that strangers don't show empathy? And we reasoned, the reason is probably stress. Stress from what? Stress from the stranger, right? You're there with this animal in an enclosed space that you don't know. Well, there could be a fight, right? They might look at you funny. And to show that indeed this was stressful, we measured stress in a number of ways shown there, and indeed it's true. Compared to being by yourself or being with a cage mates, if you're enclosed in a small space with a stranger, stress levels go up, which is not hugely surprising, is it? Okay? So if that's true, the idea is maybe stress is preventing empathy, and so then the way to show that is to block stress and see if empathy emerges. And so that's what we did, and we did it in a bunch of ways, but the data I'll show you are with a drug called metirapone, which is an inhibitor of cortisol synthesis in humans, corticosterone synthesis in rodents, and here's empathy not working in strangers again, but if they're pre-treated with metirapone, now they react just like cage mates would. And so we convinced ourselves anyway that, yeah, stress is the answer. That's what's blocking empathy in mice. And then we thought to ourselves, you know what? We could do this exact same experiment in humans. And so we did. So we got ourselves some Mogil undergraduates who are almost human. They're close. Not quite, but close. And we tested them in the pain test shown there. It's called the cold pressor test. Immersion of the arm up to the wrist in 3-degree water for 180 seconds, I think. No, more than that, 3 minutes. And at the end of the 3 minutes, they give a rating of the intensity and the unpleasantness of that stimulus, just as they take their arm out. And how this study was different from most studies is that we tested them twice. We told them we need 2 measurements, and we're going to average them so we get really good data. And half of the participants in this study were asked to come in alone, and half of them were asked to come in with a friend. We were like, oh, this graduate student, we really need to graduate her, and we really need subjects. So please bring your friend. We'll get 2 people for the price of 1. It'll be great. They came in with their friend, and then during the experiment, we made a ruse that we were behind schedule. And we said, listen, the only way to get back onto the clock, there are new people coming just in a few minutes. Would you mind if we tested you together? And we would say to them, look, you go on this end of the bath, and you go on that end of the bath. And then we told them to look straight ahead and don't talk, which meant that they were looking straight into the eyes of either their friend or a complete stranger. And then what we were interested in, of course, wasn't their ratings. We were interested in the change of the ratings from when they were tested alone to when they were tested in the dyad. Okay? And lo and behold, it works just like mice, right? If you're tested across from your friend, it hurts more than if you're tested by yourself. Okay? If you're tested across from a stranger, there's no difference. Now, I want you to think for a second just how counterintuitive this is. Your friend didn't make it better, your friend made it worse, right? And the only way to explain that is emotional contagion or empathy, which means that humans have it and mice have it, and it works the same way. But if it really works in the same way, well, we should be able to elicit empathy in human strangers by blocking stress. So how to do that? Well, we did that in 2 ways. First of all, we were lucky enough that metirapone is a treatment for Cushing's disease, so you can give it to people. And so we did. We have an experiment where they either got metirapone or placebo capsules. And then we figured, unlike mice, we could get a stress reduction paradigm in humans using psychology. And so we had them come in and play the video game Rock Band. Does everyone remember the video game Rock Band? It was a big deal about 5 years ago. You have this fake guitar, and shapes come down the screen, and if you press the right button on the guitar at the right time, a real rock song plays, and it sounds great, and you're happy with yourself. And if you play it wrong, it sounds awful. The nice thing about Rock Band is that you can play it by yourself, you can play it against another person, but you can also play it with another person. One player is the guitarist, and the other player is the bassist. So it can be played in cooperative mode. And so what we did was we had people come in, and they played, I think, 3 or 4 Beatles songs, either by themselves or with this complete stranger. And then the pain testing started. And long story short, yeah, both of these things worked just fine. In complete strangers, those that were pre-treated with metirapone showed empathy, and those that played Rock Band together instead of alone showed empathy. So again, 2 species, 2 experiments, same finding entirely. Okay? I want to give you another example of that. What I've told you about so far is the effect of social behavior on pain. But also very interesting is what is the effect of pain on social behavior. And so we did some experiments in mice and now people to look at that. So I'll start with the mice. We figured let's start off with the simplest possible question, because all social behavior requires proximity. You can't have a social interaction with someone who isn't in the same place as you. So social behavior requires approach before it can happen. So the question is, well, do mice approach or avoid pain? And I think you can imagine how it could go either way, right? So we had this little contraption set up, which is just a plexiglass alleyway with 2 jails at the end. And in jails, we put 2 mice that were either in pain or not in pain. And of course, the important thing in this study is where does the mouse that's free to roam in the middle spend its time? Okay? So we varied a lot of things. We varied the sex of the triad. We varied whether they knew each other or not. We varied whether physical contact was possible. If there are metal jail bars, they can interact with each other through the bars. But if you replace the metal jail bars with a plexiglass sheet, now they can look at each other, but they can't touch each other. So we varied that as well. And long story short, mice do not care except in one condition, right there. If they're all girls, if they know each other, and if physical contact is possible, then the mouse in the middle spends more time in the red zone. That is, more time in proximity to the mouse that's in pain. Now, again, think about that for a second. That's craziness. They don't know what's wrong with that mouse. It could be contagious. And yet, they're going over as if to help out. Does it help out? Is this social approach effective as an analgesic for the mouse in pain? With a little small adjustment to the paradigm, we looked to see if that was true. And the important data are here. If the mice are cage mates, the more contact there is with the bar, the less pain behavior, the less writhing there is of the mouse that's in pain. So the social approach works. It actually kills the pain of the mouse in the jail. And although the data I'm showing you here are from both sexes, this is sex dependent as well, because as you might imagine, female mice are simply a lot more willing to go over and touch the bar or be close to the bar than male mice are. It was then we decided, as is becoming our usual paradigm here, to see if we could do this experiment in people too. And we thought about this for a while, and we came up what I think is a devastatingly clever experiment. I can say that because I wasn't the one who thought of it. It's an experiment that's clever because the participants have no idea what the experiment's actually about. They think it's a pain experiment because there's pain testing going on. But in the middle of the experiment, they're led into a room to fill out forms. Okay? And in that room, there's a table with a bench, and sitting at one end of that table is someone who they are briefly introduced to as another participant in the study that's just 15 minutes ahead of them. But in fact, that person is an experimenter, and the experimenter has been instructed to do one of 3 things while sitting in the bench. No conversation, just one of 3 things. Either nothing, just sits there and fills out her pretend questionnaire, or making sickness behaviors, so either coughing or sneezing. This was practiced extensively beforehand so that he or she does it the same way every time. Or making pain behaviors, which I believe is massaging of the elbow or rubbing of the temple as if they had a headache. And what's really amazingly clever about this experiment is here's what we're measuring. What we're measuring is simply this. Where does the real participant sit down? Right? They have to sit down and fill out their own questionnaires, and the question is, where on the bench do they do so? How close to the person who's either pretending to be sick or pretending to be in pain? And here's what we found. If the experimenter and the participants are women, compared to someone doing nothing, they sit further away, almost 10 inches further away from people who are pretending to be sick. But they sit almost 10 inches closer to people who are pretending to be in pain. And again, if you learned anything from the mouse experiment, you would predict that there's nothing going on in men at all. Men simply don't care. They sit further away to start off with, and they don't care whether anyone's sick or in pain at all. And again, I don't think that's going to be a surprise to anyone, but the point is, once again, precisely the same finding in animals and humans. All right. So, to sum up, yeah, I know, it's awesome, eh? Yeah, I know. What I'm trying to tell you is, look, we can do translation. Things that we learn from animals can be applied to humans. It's not perfect. obviously, and it can get better, obviously, but it's clearly possible. But the irony is that while you would have expected that when you're doing biology, translation should be easy, but if you go up to sociology, it should be impossible. In fact, what we're finding is that translation works really well for sociology. It's biology where we're having the problems. And again, this is a very, very strange thing. I don't know if it's just pain. I don't know if it's just my lab. Obviously, a lot more people are going to have to start doing translation before we find out whether this is a reliable rule or not. But I find that it's particularly interesting if possible. So with that, I want to thank the folks that have been in my lab over the years. You'll notice over in the left-hand side that there's someone in the room here that used to be in my lab, and a whole bunch of undergrads and my funders, and mostly I want to thank you for your attention. So thanks a lot. Happy to take questions if there's time.
The Vitruvian mouse. I've never seen that before.
Yeah, I know. It's great. And that's exactly what it was called on that.
All right, we've got a question right over here. Just need a microphone.
Thank you very much. That was a fantastic presentation. I wanted to, I guess, clarify a point you made.
Yeah.
You're arguing that your data, your results show fairly conclusively that the psychosocial parts can be replicated more effectively through translational research than the bio parts.
Yeah.
But I think an important thing to acknowledge is that the bio results have been pooled across genders, whereas the psychosocial impacts that you're showing have been differentiated between sexes. So if we were doing a better job of doing the bio research, acknowledging that we need to do it with male mice and female mice, would that hypothesis or that claim you're making still stand up?
Yeah, look, I don't want you to take that hard and fast rule to the bank, right? It's obviously both of these statements are based on a very limited and anecdotal experience, which is just experiments in my lab working or not working, right? I don't want anyone to, it's a little bit of a sort of a fiction to make a story to hang the talk off of. So I don't want you to take it too seriously, but you're absolutely right, of course. The lesson that we've been lumping when we should be splitting is, I think, very clear in my mind, and that is my lesson to pain biologists. And in fact, I've been going around the world now for 20 years trying to convince people that yes, it's practically easier to pool. You want to pool; it makes things cheaper. If it's true, pooling generalizes better. But you pool or you lump at your own risk, because there's more and more evidence to suggest that splitting is the way the world works, that the sexes have different biologies, the different strains have different biologies, the different species have different biologies, and we simply can't assume that things are the same. You have to show empirically that things are the same. But of course, that takes longer, right? So that's where we are.
In your lab, are you starting to conduct more experiments that are female mice only?
I think if I used female mice only, I could be accused of the same, of the same thing that I'm accusing other people of, right? It seems to me that once you know there's a sex difference, once you know that the very biological structures that you're investigating might be sex dependent, I think you're morally obligated to do it in both sexes. If money was so tight that you had to say, I can only afford to study one sex, then it's very clear to me that sex should be female, right? But assuming that you have enough money to do experiments in both males and females, you're going to find more stuff. Right? You're going to turn one paper into two. You're going to turn less interesting papers into more interesting papers. There's no reason that I can think of not to do it. But that being said, I'm clearly in the minority, because people in the pain field continue to only use male mice. They're so afraid of the variability from the estrus cycle, which, by the way, has been shown to be completely swamped by the variability from dominance interactions in males. So what they're afraid of, they shouldn't be afraid of. But they're still so afraid of it and the inertia is so strong that people just continue to do what they've always done. So, we're working on it.
We have another question in the back.
Thank you. Thank you. That was a terrific presentation. Something I'd like to give you the opportunity to comment on. You spoke about empathy in mice, and in the course of talking about that, you nearly equated it, and maybe even in one instance did equate it with emotional contagion. And I am thinking that in a laboratory setting, that equation might make sense, but then in a clinical setting, we sometimes want to be very careful not to confuse empathy, which maybe has a little bit of separateness and is not emotional contagion. Could you say more about that?
So sure, and of course, if I had more time, I would have spent some time in this, because you're absolutely right. Empathy is a very controversial term. There's at least 20 different definitions that I've been able to dig up out there in the literature. And some people, most notably Franz de Waal, believe in the sort of Russian doll model that I showed. And I believe it too. I think this makes a lot of sense, that the core level of empathy is emotional contagion. That is, if you don't have emotional contagion, it's not possible to have true cognitive empathy or sympathy or pro-social behavior. You need emotional contagion first, and then these higher levels of empathy build on that. But you're absolutely right that when people say the word empathy, and when we think of empathy, and when we think of it in a therapeutic context, it's not emotional contagion we're thinking of. It's several levels above. The empirical question, of course, is whether emotional contagion truly is required for these other levels to appear. Some people believe yes, and some people believe no. I believe yes. And I also believe it's the simplest thing to study in the laboratory, and the best thing to start off studying in the laboratory, because all other forms of empathy you can only get into by questionnaire, and I don't believe in questionnaires. I believe that when people are given questionnaires, anyone with a 3-digit IQ figures out within seconds what the experiment is about, and that the only true way to study something is to fool people and make them believe the experiment's about something else entirely. Emotional contagion, you can do that. People have multiple pain testing, they know it's about pain, but they had no idea that the real dependent measure is the difference in the rating this time and that time, so I can pull out empathy without them having any idea what I'm studying. So that's why I like it. And I also think that you should walk before you run, so that emotional contagion is a good place to start. But you're absolutely right. That the real world is involving levels of empathy that are at much higher levels, are much more complicated, and the same rules that apply to emotional contagion might not apply. So I agree with you completely.
Great, thank you very much. We have to go to break now, but let's give a hand to the man that is going to help us feel more pain if we see each other in pain.